The cell cycle is a sequence of events in which the cell grows, duplicates its DNA, and as a product it divides into two genetically identical daughter cells. The cell cycle is divided into two dominant stages, which include interphase and cell division, each being split up into multiple phases. Interphase includes three phases, G1, S, and G2 phase, and is a stage where the cell prepares for cell division. Interphase makes up 3/4 of the cell cycle, whereas M phase is much shorter and only occupies 1/4 of the cell cycle. The first stage is the G1 phase, where the cells do most of their growing. During this phase, the cell grows in size and synthesizes new proteins and organelles. If the cell is big enough and the environment is suitable, the cell moves on to the S phase. The S phase is where the DNA is synthesized in the nucleus of the cell by replication of the chromosomes. Once the DNA is replicated and the cell contains twice as many chromosomes, it then moves on to the G2 phase. The G2 phase is the shortest phase of interphase, and during this phase, the organelles and molecules necessary for cell division are produced. If the DNA is replicated, the cell is big enough, and the environment is suitable, the cell moves on to the next main stage, cell division. Cell division, or M phase, which follows interphase and produces two daughter cells, is made up of two main stages, mitosis and cytokinesis. Mitosis is made up of 4 stages, prophase, metaphase, anaphase, and telophase, and is the division of the cell nucleus. The second stage, cytokinesis, is the division of the cytoplasm. The first phase of mitosis is prophase, which is usually the longest phase that could take up half of mitosis. During prophase, the spindle fibers start to form outside of the nucleus, while the chromosomes become visible and condense along with the genetic material inside the nucleus. Lastly, the nuclear envelope breaks, and the centrioles that were duplicated in interphase start moving to opposite poles while the nucleus disappears. The cell then moves on to metaphase, which is usually the shortest phase in mitosis. During metaphase, the double-stranded chromosomes line up along the center of the cell as the spindle fibers attach the poles of each end to the centromere of each chromosome. The third phase of mitosis is anaphase, which includes the sister chromatids of each chromosome beginning to separate. The centromere that holds the sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. Once the chromosomes are separated, the cell moves to the last phase of mitosis. This phase is telophase, in which the two groups of chromosomes reach the opposite ends of the cell as a nuclear envelope reforms around each group. The chromosomes uncoil and disperse into chromatin, while the spindle fiber disappears as well as the nucleolus reappearing in each daughter nucleus. This is the end of mitosis, but there is still one more stage of cell division. After mitosis, the nucleus is split into two, but during cytokinesis, the cytoplasm is split to produce two cells. Usually, it occurs at the same time as the last phase of mitosis, telophase, and it differs from animal and plant cells. Cytokinesis completes the process of cell division with two daughter cells that have an identical set of duplicate chromosomes. Cancer is when the cells in our bodies cannot control their growth, so they continue dividing abnormally. Cells receive signals that control their growth, but cancer cells do not respond to these, causing uncontrollable cell division. This is caused by faults in the genes that control cell growth and division, and as a result form tumors, which can occupy normal cells and tissues. With cancer cells, mitosis occurs much faster, affecting the cell cycle by breaking down the control of the cell has over it. Two types of genes accomplish cell cycle regulation: proto-oncogenes, which speed up cell division, and tumor suppressors, that slow down or stop cell division. The balance between the two keeps the cell dividing at a normal rate. When proto-oncogenes are mutated, they become oncogenes, which are genes that allow for excessive cell division, whereas mutated tumor suppressor genes become inactive, not allowing the cell to stop or slow down division when needed, both disturbing the balance of the cell's division, resulting in uncontrollable cell division.